Okay, again, good evening, everyone. Welcome to our Unit 2 review. I hope you all enjoyed your spring break. I know it's a little bit weird kind of coming back to this, having to take an exam so quickly. So the purpose of this review is just kind of help you recall some of the stuff we discussed earlier. It's a very heavy topic. So we'll go over some of the stuff discussed earlier a few weeks ago and really try to focus in on the more important parts, which are page 7 and page 10 of your study guide, with the physical and chemical methods of control along with antibiotics. Same thing as always, etiquette. If you have any questions, feel free to raise your hand, throw them in the chat. If there's questions that you think of later after this, or if you're watching this from home, just feel free to message me. You do have a week before our exam, so you got some time. And if you're part of the Wednesday group, you also have a little bit more than a week, but same thing, just feel free to reach out. So really quickly, why did we have this unit? What was the purpose of it? So, I like to joke around, one drop of sanitizer kills 99.9% of germs. So clearly two drops will kill all the germs, right? We all know at this point that that's not necessarily the case. But the whole purpose of this unit was how do we control microbes? How do we control them in our day-to-day lives? How do we control them in medical settings? How do we control them in other applications, either to prevent us from getting sick or what do we do after we're sick? Really the whole purpose of this unit is to be able to differentiate different types of microbes and then figure out how we're going to kill them or combat against them or defend ourselves from them. So the agenda for this is we're going to talk about a little bit about nutrition and growth for microbes. We'll talk about genetics and reproduction. And then, like I mentioned, the very big important stuff are the methods of control and antibiotics, especially because antibiotics is a little bit recent. Open the door for any questions. And then I've got a couple practice case studies y'all can study on your own. I'll kind of share the document that you could copy, and then I'll explain more about it here as we get to the very end of it. So growth phases. So recall that microbes have certain phases for growth. Everything starts in the lag phase, where the number of bacterial cells is steady or stagnant. Then you'll get the log phase, where the microbe's able to quickly start using resources around it to exponentially grow. Think like E. coli that can reproduce every 20 to 40 minutes. It's able to reproduce very quickly because it has a lot of resources around it. Log phase will continue until you get to a point where you have stationary phase. So at this point, you're starting to run out of essentially nutrients or food for the microbes. So it's essentially becoming more of like one for one. So as one bacterial cell is born, if you will, another bacterial cell is dying. So you get this stationary phase for 24 to 48 hours, and then typically after that you'll start having the death phase where there's not enough resources to keep every cell in there alive, and so they'll eventually start decreasing because they start dying off. And you all experience this in the class, especially when you come back after a week or 2 weeks. You'll see that some of our media does not look the best. That's because the media has entered a death or decline phase, or I should say the microbe in the media has entered a death or decline phase. Just know that there's different environmental conditions in which we can grow the bacteria. Um, for temperature conditions, you have different things like psychrophiles, mesophiles, thermophiles, and hyperthermophiles, and oxygen requirements. We've talked about anaerobes, aerobes, anaerobes, facultative microbes, also the aerotolerant and the microaerophiles. And then you have things like pH and salts, where you have acidophiles, neutrophiles, alkalinophiles on the pH side, and then for salt you have halophiles and halotolerant. Um, I don't necessarily expect you to have too many questions on this, where you're really going to get the majority of your questions, or if I was going to give a question on the environmental conditions, it'd be under these two specific things. So you might be given a temperature range and be asked, hey, what kind of microbe lives in this temperature range? Or it might be flipped, where I've seen it, Dr. Cotter ask, hey, what is a good temperature to grow a mesophile? And then you'd have the options of -5 to 20, 20 to 40, 40 to 80, and greater than 80 as your A through D. And then you want to pick, the best one. I have seen students struggle with this. So it seems pretty simple right now. So if you're thinking, oh, I'll remember it, just do yourself a favor, review all of these just in case the question does come up. Because I have seen students stay on this page a little too long, trying to decide whether the microbe is a thermophile, psychrophile, etc. And then for our oxygen requirements, you've seen this image before. This image was in our study guide and in our lab notes. I like 99.9% guarantee you're going to have an image like this with the letters maybe jumbled around, and you might be asked, hey, what type of microbe is D, for example, or B? This one for facultative, this isn't the exact image you're going to use. Just remember that facultative has growth all around. This internet image doesn't do the best job of showing that, but the image on the exam will be better than this. Fermentation. So this is also another question that I've seen students miss. Typically, it'll ask you like, hey, what are the three three products of fermentation? And I've seen alcohol, I've seen CO2 gas, acid. Those are the main three ones. I've seen some people mix stuff. Well, they'll say like alcohol gas, or I've seen people just only get two of the three. Uh, Again, this is one of those like great short answer questions where you just get asked, hey, what are three products? And you'll put alcohol, CO2 gas, acid. Quick 3 points, or should be a quick 3 points. So again, if you don't remember these, just refresh them. You've got a week to do them. It seems very simple, but it seems like during the exam time, people will sometimes forget one or two of these. Water movement. So water movement, we typically talk about different types of cell conditions. And so if you're taking any of my Bio 201 or 202 classes, similar to my Bio 202 class, we'll talk about how water follows salt. And so the cell will either become dehydrated, it'll be the right size, or it'll be too water heavy, depending on where the salt is. So a hypertonic solution means that there's more salt outside of a cell than there is inside the cell. And so water inside the cell will leave the cell to try to balance itself out with the salt around it, and it causes the cell to shrink. A hypotonic solution means that the cell is more salty than the water around it. And so water will make its way into the cell to try to balance out the sodium concentration. And eventually the cell will swell up and it'll lyse or it'll burst. And an isotonic solution, that just means that you're balanced salt inside of a cell and outside of the cell. So there's no water movement and the cell size does not change. Again, if you had a question like this, it's probably going to be one question. I wouldn't worry too much about studying this portion. I just wanted to kind of refresh it. Genetics. I think you'll probably get, if I was giving the test, I'd probably give like two questions on this. You saw it in the case study where we gave you a scenario and we asked you which of these could it not be because it has a sex pilus, and you're like, okay, conjugation has sex pilus. So as you look at the three different types of horizontal gene transfer, this specifically for bacteria, we've got our conjugation, our transformation, and transduction. Conjugation keywords here are the sex pilus, and that you're copying a plasmid from one cell to another, and this allows the new cell to acquire antibiotic resistance, and also to develop its own sex pilus so where it can then share its information to other microbes. Transformation, this is what we try to do with the P-glow plates, where we are trying to essentially get a piece of DNA fragment. In our case, it was a plasmid, but it could also just be other fragments of DNA. And we're trying to integrate it into an existing cell. And then we're hoping that those fragments become part of the cell. And now this new microbe, this new cell of the microbe has new types of information that it could be used to change itself to survive. I don't know if it was this class or my Wednesday group, but I mentioned how this is a way for us to make antibiotics. So we'll actually take a plasmid with a certain DNA structure for an antibiotic like amoxicillin, and then we use transformation to get that plasmid into an E. coli cell, and then we mass produce these E. coli's to mass produce the antibiotic for us. And then at the very end of it, once we get a certain amount of bacterial cells, we'll essentially go through a process of destroying or killing all of the bacteria and then spinning them down to where all the cell parts, which are heavy, go all the way to the bottom. And then we extract the antibiotic. And then we can package the antibiotic and sell it to a pharmaceutical company. Essentially, we're using E. coli as our factory to mass produce antibiotic, which is a pretty cool thing. I've also done this with plants where I've taken a plant, essentially infected it. It was kind of a combination of transformation and transduction, but essentially we infected it with a virus, and this virus had a piece of antibiotic and a red fluorescent protein, and we injected it into a plant. And essentially, this virus overtook the plant to where the plant was constantly mass producing antibiotics at a cost of its own. So it actually was killing the plant slowly. But for the next like 5 days or so, we were able to use this plant to mass produce antibiotic. And then we did the same thing. We took the plant, we kind of shredded it into pieces, extracted the antibiotic, and then we shipped off the antibiotic to a pharmaceutical company. So a lot of things that are you're kind of looking at like, oh, we did this in lab, but what does this all mean? Well, this is actually a way that we use today to mass produce antibiotics for us. And then transduction. Transduction is something that involves a bacteriophage or a virus where the bacteriophage accidentally takes a bacterial DNA piece instead of a viral DNA piece, and it transmits it to another cell, and then that cell gains the bacterial DNA information to potentially develop antibiotic resistance. I think for the exam, like I said, you might have images like this, or you might be asked more in words like, hey, what does transformation do? What does conjugation do? That kind of thing. Interject drift versus shift. This should be fairly straightforward. Drift is a small or minor change to the viral spike protein. It is a slow change and it involves point mutations. Those are your keywords for antigenic drift. And antigenic shift, it's a major change in the viral spike structure. It's a very fast or quick change, and it's caused by genetic recombination. Um, if you have a question that's talking about influenza, hint, hint, you might be also asked a question about if it's drift or shift, and why is it drift or shift. So just just study that just in case you see it. Okay, so like I said, I didn't want to really spend too much time on the other stuff because for the most part, they're going to be like onesie, twosie questions. Where you're really going to be uh, heavily tested on is going to be page 7 and page 10 of the study guide. And that is your physical and chemical methods of control, and then also your antibiotics. So uh, it is a very long list, and sometimes we get a little bit overwhelmed with the amount of stuff that's on there. So I've been telling students to do for the last 6 years now is when you study for this test, try not to focus on learning everything or memorizing everything on page 7. Instead, pick one or two methods that'll do, two methods from physical and two methods from chemical that sterilize. And that way, if you see a question that asks you, hey, how do you chemically sterilize this? You just go to your one or two that you've selected, and you have to worry about the rest. So when you go through here, physical methods of sterilization, you're going to have autoclaving at 121 Celsius at 15 psi. You're going to have UVC at 260 nanometers, and you'll have filtration at 0.025 micrometers. If you just memorize two ways to sterilize, you can answer basically most of the questions Dr. Coward is going to ask. So don't focus on all of them. Just pick one or two that you like, that you'll memorize, and that way you go with those on the exam. So for physical methods, you've got your two ways of sterilizing. Then pick two ways to disinfect. Either boil, I picked boiling, pasteurization, dry heat. You can pick two that you like. If you want to do three, you can. I think for the most part, I've seen as long as you have two, you have your primary and your backup just in case you forget your primary, you're going to be fine. The way the questions are going to be phrased, and we'll kind of show you this on the practice cases, is that you're going to be given the case study. You're going to be given this microbe or whatever, and it's going to ask you, what is one physical method that you can use to sterilize this microbe? And you're like, bam, autoclave. And then the second question might be, give me two ways we can physical methods to disinfect. Okay, I got boiling and pasteurization. Next question. Okay, give me a chemical method, or what are the three chemical methods that we can use to sterilize? For sterilization for chemicals, you should know Oz, the aldehydes, halogens, and hydrogen peroxides. Hopefully, these are the three I would memorize because the question could actually ask you what are the three ways that we can chemically sterilize. But for the most part, you might also be asked the way like, hey, pick one or two of these for this microbe. And the fourth question, hey, what is one or two ways we can disinfect, chemically disinfect? And if you sum up the way I just phrased it, that's already 8 to 10 points on just you answering a physical way to sterilize, a couple ways to disinfect, a couple ways to chemically sterilize, a couple ways to chemically disinfect. This is usually where students struggle because they mix them. So they'll say for sterilization, I put boiling, or for my chemical, I put bleach, or they mix the chemical and physical, where it's like she's asking for a physical method of control and you give her a chemical. You're going to get that wrong 100% of the time. So try to just get your two per row, two sterilants, two disinfectants, two antiseptics for physical, and then two chemical sterilants, two disinfectants for chemical, and two antiseptics for chemical, and roll into the exam with that, and you'll be I'll be pretty confident that you'll be good to answer most if not all of the questions. Um, and then for antibiotics, I have the exact same strategy. So page 10 has a lot of antibiotics. Um, the way that you want to break them down is the way we broke them down in lecture today, or the way you'll break them down in lecture on Wednesday, is that for the plasma membrane, we really only have one drug that targets the plasma membrane. So memorize that one. Peptidoglycan cell wall, I think we had a list of like 6 or 7 of them. Don't remember all 7 of them. Pick 2 of them. Anything that ends in cillin, you can go after a peptidoglycan cell wall. So ampicillin, amoxicillin, penicillin. If it ends in cillin, peptidoglycan cell wall. Then you'll have, 1 or 2 for the waxy cell wall. Pick a folic acid metabolism drug and know which one is more or less toxic, because the scenario might be asking you, hey, which of these is less toxic to the patient? Another way I've seen the question worded is, which of these is more toxic to the patient? And so make sure you know toxicity levels, which is less or which is more. Protein synthesis, again, get 2 or 3 of those drugs and just know which one is less toxic and more toxic. Ideally, you're always giving the less toxic version to your patient. Pick a DNA inhibitor, and then know your antivirals. There's only really 3 of them that you should worry about. Pick one for influenza and pick one for HIV, and you should be all set. Does that make sense to everybody in the call? Just kind of how I'm having you do it. Dr. Codd had a good idea for, take a drug and then on the opposite side, write down like what it targets as like a flashcard setting. You can do that. I don't really use flashcards myself. I just kind of decided like I'm going to focus on just studying, 8 drugs versus the entire list of drugs, if you will. Okay, that's really all I had for the knowledge portion. Does anybody have any questions while we're in here? Anything that I can answer for you? If not, I'm going to open up the practice cases and I'll kind of show you how the practice cases are going to work. And if you have any questions while we're talking, you can also just chime in. Okay, perfect. You should be seeing the practice cases now. So I'm going to copy this, put it in the chat. And I'll also include this link in the announcement with the video link for those that are watching remotely. Can you show the page with transduction? Yes, I can. If you want to take a picture of it, you can. Where'd it go? This one. There we go. Okay, let me start the slideshow. See if it'll let me. Okay, go ahead and you can go ahead and take the screenshot. Okay, perfect. No problem. Okay, so like I said, I gave you guys a link for the practice cases. I will share this in the announcement. So these practice cases are typically the ones that Dr. Cotter shares with y'all in class. Over the last few years, if we've separated it to where we do them during the exam review, mostly because everyone would kind of just jump in here and start typing up a bunch of stuff, and they weren't actually able. It was kind of like everyone was getting in everybody's way. It didn't really help for students to learn. So the link here, you'll see it's read only. You have to go to File, make a copy, and you can make yourself a copy that you can edit. And so the way I've phrased, we have these is I've given you three scenarios with three different bacteria. So I gave you Mycobacterium tuberculosis in a surgical room. Another scenario, endospore forming bacteria like Clostridium difficile in the GI tract of a patient. And then a third scenario of Pseudomonas aeruginosa in a water system, i.e. air conditioning or HVAC of a hospital room. So when you're answering these, take page 7, take page 10. I started you off here with like, kind of gave you a quick answer here, but it's like, okay, Mycobacterium tuberculosis, what type of microbe are you trying to treat? That's the first question you should ask yourself in all of these case studies on the exam. What type of microbe is this? Is this gram positive, gram negative? Is it an endospore former? Because depending on what your answer is, that will determine how you're going to sterilize it. How are you going to disinfect it if you can? I.e., an endospore cannot be disinfected, it has to be sterilized. And also what chemicals you can use and what drugs you can use to attack it. So I just gave you this acid-fast bacteria with a waxy cell wall. That's a hint, right? Waxy cell wall. Only one drug targets waxy cell wall from what I remember. I'll let you figure that out on your own. And then you're going to go and list physical methods that we can use to decontaminate, physical methods that we can use to sterilize, physical methods used to disinfect, chemical methods used to decontaminate, the Oz, and then high-level disinfectants, chemicals, high-level chemical disinfectants. I'll do that. And then chemical methods for antisepsis on living surfaces, i.e. on a person. Also know which ones are residual and not residual. So I gave you guys little reminders. Residuals sticks on the surface and non-residuals evaporate. I have seen some questions that do ask specifically like, hey, you want to leave a residual chemical on this surface. What chemical should you use? And then the last question is, what drugs are you going to use to treat tuberculosis or this acid-fast bacteria? And write your one or two here. And then you'll just go through and answer all of these. Again, this is for your own practice, so feel free to do this at home, whatever it might be. Whenever you're done, whenever you get, you're done or you get kind of stuck or you're kind of ready for some feedback, I'm going to treat these like the case study, so you can feel free to message me and just send me the link to your Google Doc, and I'll go through them and I will kind of let you know, like, yeah, you're good, or like, oh, actually, this does not go here. This goes somewhere else. You should go and add these things instead. At least what I heard from the last semester's class, the students that actually did this and answered this fully, I saw them do much better on the Unit 2 exam than the students that did not attend the session to get this notes or they didn't perform it. Mostly because some of these might actually reappear on the exam. I don't know. Dr. Trotter has a revolving exam, but some of these might be very similar on your exam to where you remember like, oh, I remember this drug or I remember doing it this way. But overall, it's just a good way to practice how the cases are going to look on the exam. And if you look at how it's written, this is one like bacteria, and typically gave you basically gave you 8 questions. That's a good chunk of your exam. And if I give you 3 or 4 of these, that's 24 questions, 22 to 24 questions. That's a good chunk. That's, let's say, about 50% to 60% of your exam. I have a couple things in the chat. Um, okay, gotcha. Yeah, yeah. I think I answered your question, but yes, this is not for points. This is straight for you to practice cases that are very similar to what you'll see on the exam, especially the wording. I want you to really practice, like, make sure you're not putting chemical disinfectants in the physical method sterilization section. Really pay attention to the keywords because the keywords like surgical room or things like GI tract. So that tells you that's inside a patient. You probably want to give them something that's not toxic, just, as an example. Those are the keywords that are going to tell you whether you picked the right sterilants, the right disinfectants, and also the right drugs, if you will. And like I said, like a case study, feel free to work on it and then send them to me and I will review it. And then the case study, yeah, so the case studies for this unit, I think that's what you're referring to, are going to be also going to help you on the exam. So if you want to review the case studies from like chapter 11, specifically chapter 11 and chapter 12, those are the ones you want to really focus on um, as you prepare for the exam, because you'll also have very similar cases like the ones I'm showing you here. This one just kind of puts everything together with the types of microbes, the methods, and then the antibiotics, which is how your exam is going to be worded. Okay, that's really it. So 30 minutes, not too bad. Again, I think the biggest highlight or biggest takeaway I want you all to have from this is don't stress out about remembering or memorizing all of page 7 and page 10. Pick a few chemical methods of control that are sterilants and disinfectants. Pick a few physical methods that are sterilants and disinfectants. Pick a few drugs that target different parts of a cell and study those. And for the most part, you're going to do just fine on the exam as long as you've got your primary drug and your backup drug, for example. Or if there's only one drug available, then make sure you memorize that one drug for that portion. Um, that's all I had. That's all I had. Does anyone have any last minute questions at this time? And if I don't have any questions, I'm going to again say thank you very much for your time. Feel free to rewatch this if you want. And then, like I said, message me with the practice cases. I'll review the practice cases up until Sunday. So I know the case study is due on Friday. So if you want to get that one done first and then work on this during the weekend, you can do that. You can message me. I will probably stay up until like 10 p.m. on Sunday, maybe 9 p.m. So long as I have them before then, I'll at least give you some feedback before I lay down and see you on Monday. Other than that, have a good night, everyone. Enjoy the rest of the week, and I will see Monday group. I'll see you on Monday for the exam. Wednesday group, I will see you on Wednesday.